Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International GCSE Biology Paper 2B for November 2023. Let's begin with the first question. Question 1. Read the passage below. Use the information in the passage and your own knowledge to answer the questions that follow. The passage is about plants to the rescue. They say since the early 20th century, the concentration of carbon dioxide in the atmosphere has increased rapidly. This has further increased in recent years due to more cars and the increased demand for electricity for homes and industry. Scientists have found that plants play a critical part in removing this excess carbon dioxide from the atmosphere. Using computer models, the scientists concluded that photosynthesis has increased by 30%. The scientists measured carbonyl sulfide found in ice cores and air samples. In addition to carbon dioxide, plants take in carbonyl sulfide gas during their natural carbon cycle, and this is frequently used as a measure of photosynthesis on the global scale. Terrestrial plants are removing about 29% of carbon dioxide emissions that would otherwise contribute to an increase in the atmospheric carbon dioxide concentration. A carbon sink is an ecosystem, such as a forest, that absorbs more carbon dioxide than it releases. The size of the carbon sink depends on the rate of photosynthesis, but also on the level of deforestation and respiration. The model that scientists used showed that the role of photosynthesis in producing a carbon sink in non-plants is larger than estimated in most other models. Other scientists are less confident about using carbonyl sulfide as a measure of photosynthesis. Plant absorption of carbonyl sulfide can vary depending upon the amount of light that plants receive. Therefore, the measure of global photosynthesis could be overestimated. Regardless of the rate at which photosynthesis has increased, scientists agree that excess carbon dioxide is boosting the growth of plants. Trees are becoming leafier and there is more wood. The wood is where most of the carbon is stored in the plant. In experimental research, scientists expose plants to double the normal concentration of carbon dioxide found in the atmosphere. Under these increased carbon dioxide conditions, the composition of the leaf tissues changed. This made the leaves tougher for herbivores to eat and made it harder for insect larvae to grow. Scientists have also observed that when plants are exposed to increasing levels of carbon dioxide, the size of the stomatopores of the leaf increases. So the first question says, explain why more cars would result in an increase in atmospheric carbon dioxide. This is because cars use petroleum and diesel, and these come from fossil fuels, and when these are burnt, you produce carbon dioxide, which increases the atmospheric carbon dioxide concentration. And in part B, explain how increased carbon dioxide can cause climate change. Because CO2 is a greenhouse gas, greenhouse gases trap infrared radiation, and then they re-emit it back onto the surface of the Earth, causing the global temperatures to increase. This is what we call global warming. So when global warming occurs, it can lead to the melting of the ice caps. This could cause flooding, increase in sea levels, and so on, and this is what we call climate change. In part C, explain why increasing carbon dioxide concentration can increase photosynthesis in plants. During photosynthesis, carbon dioxide is a reactant. Remember, the reaction for photosynthesis is carbon dioxide plus water gives us glucose plus oxygen. So CO2 being a reactant, when its concentration is increased, the rate of photosynthesis is going to increase because the plants are going to be taking in more CO2, which is a reactant. However, this could level off if another factor limits the rate of photosynthesis. For example, the graph could look like this. Initially, it's going to increase, but it could level off if some other factor is limiting the rate of photosynthesis. And in this graph, we'll have carbon dioxide concentration on the horizontal axis, and we will have the rate of photosynthesis on the vertical axis. So at that point, when the leveling off begins, no matter how much we increase the concentration of CO2, the rate of photosynthesis will not increase because another factor is limiting the rate of photosynthesis. In part D, explain why the carbon sink depends upon respiration and deforestation as well as photosynthesis. Photosynthesis is a carbon sink process because it uses up carbon dioxide from the atmosphere, fixing it to produce glucose as well as other carbohydrates that could be stored within the plant. However, respiration will use up those carbohydrates, producing carbon dioxide, and respiration will increase the atmospheric carbon dioxide, while photosynthesis is going to decrease it. However, if deforestation has been carried out, it means we have less trees that can carry out photosynthesis. There is going to be an increase in the concentration of carbon dioxide because the carbon sinks have been decreased. So I said photosynthesis is a carbon sink process because it uses CO2. However, respiration produces CO2, meaning it increases the CO2 concentration in the atmosphere. Also, deforestation decreases the uptake of CO2 and increases the atmospheric carbon dioxide through decay and decomposition, while others could be burnt down, and combustion processes also increase the CO2 concentration. In part E, light affects gas exchange in leaves. Describe an experiment that shows how the net gas exchange of a leaf varies with light intensity. So there are two ways this experiment could be carried out. One could be using a water plant, and then you count the bubbles of gas that are produced. And another could be carried out using a hydrogen carbonate indicator, like I described here. So I said you set up test tubes, include a hydrogen carbonate indicator solution in each, then place a leaf in each test tube, place one test tube with a leaf in the dark and another in light. The color of the hydrogen carbonate indicator solution will turn purple for the experiment in the light, and it will turn yellow for the experiment in the dark. However, when you carry out this experiment, the leaves used should be the same number, they should have the same surface area, they should be the same age. If possible, they should come from the same tree. And the hydrogen carbonate indicator solution should have the same volume as well as concentration. Also, the experiment should be carried out for the same period of time. Now, if another person used a water plant, you set up an experiment with different light intensities, meaning you could use like 5 different light intensities. The different light intensities are maintained by varying the distance of the lamp from the experimental setup. Then you count the bubbles of gas produced after some time in order to calculate the rate. You could repeat at each light intensity and then calculate the mean, and then you compare the results from the means at each different light intensity. In part F, the photograph shows stomata on a small square of the lower surface of a leaf. So we can see we have 2 stomata, there is one and the second one. And here they've given us this is a square which is 0.4 millimeters. The total area of the lower surface of the leaf is 150 centimeters squared. Using the photograph and the total lower surface area of the leaf, estimate the number of stomata on the lower surface of the leaf. So because they've given us this area, I'm also going to use these measurements here and find the area of this part of the leaf. The area is going to be the side length squared, which is 0.4 squared. However, this is going to be 0.16 millimeters squared. But this is in centimeters squared, so I have to convert the centimeter squared into millimeters squared. Since 1 centimeter is 10 millimeters, 1 centimeter squared is going to be 100 millimeters squared. So 150 centimeters squared is going to be 150 times 100, giving us 1,500 millimeters squared. So in this area, which is 0.16 millimeters squared, we have 2 stomata. The area 1,500 millimeters squared will contain 2 divided by 0.16 times 15,000 stomata, and the answer is 187,500 stomata. In part G, stomata also have a role in water transport in the plant. Explain the role of stomata in water transport. This question is focusing on water transport, so we're going to talk about transpiration as well as movement of water through the xylem. So I say stomata enable water loss to occur through the leaves by transpiration. This then draws water from the roots through the xylem. This is in the transpiration stream. So this brings us to the end of question 1. Let's continue to question 2. In question 2, catalase is an enzyme found in many cells. This enzyme controls the breakdown of hydrogen peroxide into water and oxygen. So this is the reaction for that breakdown. And they say a teacher uses this method to investigate the effect of pH on catalase. Again, remember catalase is an enzyme. which carries out this reaction. They say cut a cylinder of potato tissue into 6 equal-sized discs and then measure 10 centimeters cubed of hydrogen peroxide solution and place into a boiling tube. Add a buffer solution to the test tube to keep the pH at 7. Add the 6 potato discs to the boiling tube. Collect the gas given off by the reaction in an inverted 20 centimeters cubed measuring cylinder. Then measure the total volume of gas collected after 5 minutes and repeat steps 1 to 6 using 4 different pH buffers. There is pH 4, 5, 6, and 8. The diagram shows the teacher's setup. So we have the hydrogen peroxide here with a buffer to maintain the pH, and then we have the potato discs. We can see they're broken down into smaller pieces so that the surface area is increased, since the enzyme catalase is leaking out of the potato into the solution of hydrogen peroxide for the reaction to occur. And here we see oxygen gas, which is a product, is collected over water in an inverted measuring cylinder. So the first question says, state what is meant by the term enzyme. An enzyme is a biological catalyst and its purpose is to speed up the rate of reaction. In part B, suggest why each potato cylinder was cut into 6 discs rather than left as one cylinder. Because the potato cylinder contains the catalase enzyme, when we cut it into smaller discs, we are increasing the surface area so that more enzyme leaks out to come into contact with the hydrogen peroxide for the reaction to occur. So I say to increase the surface area for greater interaction with hydrogen peroxide for a faster rate. And in part C, give the dependent variable in the experiment. The dependent variable is what we measure, and in this case we are measuring the volume of gas produced or released in 5 minutes. The teacher controls the time for gas collection and also the volume of hydrogen peroxide used. State 2 other abiotic variables the teacher should control in this experiment. Because this experiment involves using enzymes, we need to control the temperature, we need to control the pH, the concentration of hydrogen peroxide, and more. In part D, they say the table shows the teacher's results. So these are the experimental results. We can see the pH from 4 to 8, volume of oxygen collected after 5 minutes. We can see it increases and then it goes down. The highest volume of oxygen is collected at pH 7, and that's where we have the highest rate. So it tells us the optimum pH is closer to pH 7. It could be between pH 6 to 7 or between pH 7 to 8. So they say calculate the percentage change in the mean rate of reaction as the pH is changed from pH 4 to pH 7. At pH 4, the mean rate is 0.8, and at pH 7, the mean rate is 2.4. So the change in the rate is going to be 2.4 minus 0.8, giving us 1.6. And so the percentage change is equal to the change divided by the original rate, which was 0.8 times 100, giving us 200%. Next, they say the teacher calculated the mean rate of reaction by collecting the oxygen released for the first 5 minutes of the reaction. Explain why the mean rate of reaction changes beyond the first 5 minutes. The teacher only collected for the first 5 minutes because the initial rate is going to be the highest rate. However, as the reaction proceeds, more hydrogen peroxide is going to be broken down and the rate is going to slow down. So I said after the first 5 minutes, the substrate, which is hydrogen peroxide, is used up. So fewer collisions or no collisions occur between the enzyme and the hydrogen peroxide, and the rate is going to be lower. Next, they say explain the effect of changing pH on an enzyme-controlled reaction. And again, here they've said change, not increase or decrease. It can be any change. So I said enzymes work best at their optimum pH. Changing the pH from the optimum changes the shape of the active sites. This means it denatures the enzymes. So the enzyme and the substrate will no longer be complementary and the rate of reaction is going to decrease. So this brings us to the end of question 2. Let's continue to question 3. In question 3, the diagram shows a nephron from a human kidney with some structures labeled. The glomerulus, the Bowman's capsule, we see the PCT or the first coil tubule, we see the loop of Henle, the second coil tubule or sometimes called the distal convoluted tubule, and then we see the collecting ducts. So the question says, which structure is the Bowman's capsule? This is the Bowman's capsule, so structure S, and therefore C is the answer. The next they say which structure is the loop of Henle. The loop of Henle is going to be structure Q, so B will be the answer. And then they say which structure is affected by ADH. ADH affects the collecting ducts as well as the second convoluted tubule, so I'm going to go with the collecting ducts structure P, and A will be the answer. In part B, blood plasma contains much glucose, but urine normally does not. Explain what happens to glucose in the kidneys. In the kidneys, glucose is going to go through the glomerulus and then it's going to be filtered through to the Bowman's capsule space. So I said glucose is filtered from the glomerulus into the Bowman's capsule space under high pressure. This is through the process of ultrafiltration, and 100% of the glucose from the glomerulus is going to go through to the Bowman's capsule space. And then in the first coil tubule, or what we call the proximal convoluted tubule, the glucose is 100% selectively reabsorbed into the blood vessels by active transport using ATP. So here keywords are selective reabsorption, active transport using ATP, as well as what occurs in ultrafiltration. And next they say describe how a sample of urine could be tested for glucose. Since glucose is a reducing sugar, we use Benedict's solution. The solution is going to change from blue to either green, yellow, orange, or red based on the concentration. If it turns red, then there is a higher concentration of glucose. And if it turns green, then the concentration of glucose is going to be lower. So observation of any of these four colors would mean there is a reducing sugar, and in this case, it's going to be glucose. And in part C, they say, as a person sweats, they may become dehydrated. Describe the changes in a person's urine if they become dehydrated. If they become dehydrated, the urine is going to be less in volume and it's going to be highly concentrated. And this is because more water will be reabsorbed by the body to overcome the dehydration. So this brings us to the end of question 3. Let's continue to question 4. In question 4, mutation can cause changes in the enzymes produced during protein synthesis. State what is meant by the term mutation. A mutation is a random change to DNA. And in part B, they say explain the different effects that a single base mutation can have on transcription and translation and the production and activity of an enzyme. Now, if there is a single base mutation, of course, transcription will occur to produce the messenger RNA, but the produced messenger RNA will have a codon that is different from the way it should be. So the messenger RNA will leave the nucleus to the ribosome to be translated in order to produce the polypeptide. However, the transfer RNA that should bring the amino acid is also going to have a different anticodon, meaning the amino acid brought in this position is going to be different from the amino acid that would have been brought if there was no mutation. So the produced protein could be different because the amino acid sequence was different or is different, and so this protein may not have the required active sites complementary to the substrate, and so it will not be a functional protein. However, there is also a chance that the mutation will have no effect, because if the mutation occurred in the third base of the codon, then there is a chance that the amino acid that is going to be brought is going to be exactly the same. The produced protein could have the same active sites, have the same 3D structure, and therefore be functional. In part C, scientists investigate the rate of spontaneous mutations in yeast. They record the number of mutations per cell during mitosis and during meiosis. They count the mutations in 3 different strains of yeast and repeat each count. The mutation rates are counted per division per cell. So we can see these are the results. We see the different strains. They have 1, 2, and 3. And in mitosis, we see the first count and the repeat for this strain, the first count and the repeat for that strain, and the first count and the repeat for this strain. In meiosis, we see the same thing, the first count, the repeat for each strain. In the results, we can observe that in mitosis, this was the highest. This is where we had the highest mutations in this strain, and this strain here had the lowest mutations. While in meiosis, this strain here had the highest mutations, and this strain here had the lowest mutations. Overall, we see the greatest difference in mutations between mitosis and meiosis occurred within strain 5207, and the lowest difference is going to occur between this strain 5209. These are going to have the lowest difference. The first part says calculate the mean mutation rate for strain 5207 during meiosis. 5207 during meiosis, we have that and that, so this plus that divide by 2. But remember, this is times 10 power 8 per cell division, so this is what I got, times 10 power 8, and this was my answer to 3 significant figures. And next I say suggest why the mutation is calculated per division per cell rather than just per cell. This is because mitosis goes through a single cell division, while meiosis goes through 2 divisions or 2 cell divisions, so the results will be inconsistent, meaning if we counted the 2 divisions as 1, there could be a very big difference between the number of mutations in meiosis and mitosis. And next I say comment on the differences in rates of mutations for mitosis and meiosis in the different yeast strains. Every time they ask a question about comment, you need to describe the data, talk about the methods used, talk about the sample size, and so on. But in this case, they're asking us to comment on the differences, so there is no need for sample size and there is no need for methods used, so I'll just describe the data. I said meiosis has more mutations per division in all yeast strains. The highest rate for meiosis is 132.6 times 10 power 8 in strain 5207, while the lowest is 1.42 times 10 power 8 in strain 5209. The highest for mitosis is 6.53 times 10 power 8 in strain 5160, and the lowest is 0.21 times 10 power 8 in strain 5209. The repeat experiment per strain show a significant difference in the results, mainly for strain 5160. So this brings us to the end of question 4. Let's continue to question 5. Question 5. The diagram shows the nitrogen cycle with some processes labeled. So here we have nitrogen in the atmosphere, nitrogen fixation into nitrogen in organic molecules, and then of course nitrification to produce the nitrates, and then denitrification to convert it back to gaseous nitrogen. This explains why plants need nitrate ions. Nitrate ions are needed to make amino acids, and these are used for making proteins, for growth and repair. Also, nitrates are used to make nucleotides, for DNA and RNA. The next they say name the process V, X, and Y. Like I've already written there, V is nitrogen fixation, X is nitrification, and Y is denitrification. In part B, scientists measure the dissolved nitrate level in a river that passes through farmland. They also measure the nitrogen compounds being used on the farmland either as fertilizer or as manure from livestock. The graph shows how the nitrate levels in the river and the nitrogen compounds applied to the farmlands changed from 1950 to 2005. On the vertical axis, we see the nitrate level as well as the nitrogen compounds, while on the horizontal axis, we have the time in years. And we can see both the nitrate level in the river as well as the nitrogen compounds in the soil increase over time. But after 1970, we begin to see some oscillations within the nitrogen compounds in the soil, while the nitrates in the river are continuously higher and increasing. This could mean that the nitrates in the river are not solely due to the nitrogen compounds in the soil leaching into the river. There could be some other factor responsible for the increase in the nitrates within the river. Next, I say, Comment on the relationship between nitrate levels in the river and the nitrogen compounds applied to farmland. Here I'm just going to describe the data. So I said from 1950 to 2000, both nitrate levels in the river and the nitrogen compounds applied to the farmlands increased. After 1970, the nitrogen compounds added to the soil have fluctuations, but no fluctuations in the nitrate levels in the river. The nitrate in the river is due to leaching of nitrate from the farmlands where fertilizers are used, and the lack of fluctuations in nitrates in the river when fluctuations occur in the soil nitrogen is because there are other sources of nitrates in the river. Describe the changes within the river that would have occurred between 1950 to 2000. Here they're asking how is the river going to change. Of course, eutrophication could have occurred. So I say due to leaching of the nitrates, more algal bloom would result. There will be less light or sunlight into the water for photosynthesis of the water plants. I'm talking about the plants under the water, not the algae on the surface. So there will be more decomposition by aerobic bacteria, and this could deplete the oxygen. So the water organisms could die due to lack of oxygen. And finally, there will be a decrease in biodiversity within the river. So this brings us to the end of question 5. Let's continue to question 6. In question 6, describe how stem cells are different from other cell types. Stem cells are unspecialized, meaning they can be used to form different types of cells, and stem cells can divide continuously, producing other stem cells if specialization has not occurred. In part B, they say stem cells from a donor can be used for stem cell therapy. Human blood cells are produced in bone marrow. Explain how using stem cells from the bone marrow enable doctors to treat a number of different blood conditions. We know blood cells are made from bone marrows, like red blood cells, white blood cells, platelets, and so on. So if bone marrow transplants occur, it means we can treat blood conditions like anemia and so on. So I said the bone marrow make blood cells like red blood cells, platelets, and white blood cells. Bone marrow transplants provide stem cells that can form all different blood cells. Anemia can be treated using red blood cells, so the blood will have sufficient oxygen. Platelets can be used to treat blood clotting problems, while white blood cells can boost the body's immune system against infection. In part C, they say, one type of stem cell transplant uses a patient's own stem cells. They can be used if the patient is producing enough healthy bone marrow cells. These cells can be collected, frozen, and stored for later use. Explain the advantage of using a patient's own stem cells rather than using stem cells from a donor. If the patient's own cells are used, there will be no rejection because the body's immune cells as well as the donor cells will contain the same antigens, so the immune system will not reject the donated cells. Also, there will be no need to take immunosuppressant drugs. Also, since they are from the same patient, they contain the same genes, meaning the genes that are used to make these antigens. So this brings us to the end of question 6 as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.